fatty, semi-fatty, OGSB cookie, figured I'd do a pretty cool episode for you guys. Trying to do a series here about the plant, getting into a bunch of really cool aspects about the plant that we may not be aware of. There's a bunch of really cool stuff here that we'll be getting to in the future. As far as like how plants actually absorb CO2, and a graph of obviously how the nutrient uptake at different pH level. These are like how the soil particles interact with different nutrients and stuff like that. So that's all stuff I'm going to be getting into, but we kind of need to understand how the cells work. If you're interested, this is probably going to be the most informational of how cells actually work and how we're actually able to function as human beings. Plants function as plants. As you see, this is a plant cell right here. The green things in there are chloroplasts, obviously. Most of you probably know that. And then this other little thing, this little kidney-shaped thing in there, that's the mitochondrion. And, this is another very, very important part of the cell as far as producing ATP. It is the most important energy source of the cell. And I thought it was kind of funny about the plant ATP. ATP actually stands for adenyl triphosphate, I believe. And phosphate is used within the entire cell as far as it's like involved with energy production. Phosphorylation is involved with putting phosphates on things, usually proteins or enzymes, and it makes them more active. And then dephosphorylation, obviously taking the phosphates off of them, making them less active. And this happens a lot inside the plant cell, inside our own cells. These two energy sources are very important, keep in mind, but we're going to focus more on the nucleus right here, which houses the DNA, or well, does encode for all function of every living organism. Physical structure, internal structure, everything that we see, do, have in our body is encoded for in DNA within this nucleus, within every single cell within our body. There is an amazing amount of nucleic acid. Nucleic acids have a sugar group right here. They have a phosphate group. These are the 4 nucleotides that make up all of DNA. The base, the guanine, they have the cytosine here, the adenine, and the thymine. These 4 nucleotides code for the entire DNA sequence. See on the right side here, G, T, G, A, T. That's a sequence. Now, if this were encoded for in a gene, the TAG, that would be a single amino acid. I wanted to explain how all of this genomic material fits inside this nucleus. I think in humans it's like 6 miles of DNA. This is from plant genetics. Flowering plants have the largest genome, including mammals. We're like a 16th of the size of the genome of plants. So it's pretty amazing. Anyway, DNA is wrapped around in a helical structure like this, E form, the beta form. You can see the nucleotides within the sugars and phosphates on the outside. They interact with water a lot better than internal bases. All of the DNA is wrapped around these histones, wrapping the DNA very, very, very tightly, and it ends up becoming a chromosome. And these histones actually have tails on them. Histone tails, they're made of amino acids. The reason that they have tails on them, represented over here, is so the DNA can be moved around, and it's moved around by these remodeling complexes right here in green, and as you can see it involves ATP, and this is moving the DNA and allowing proteins to come in and replicate the DNA. The replication of DNA involves many proteins, and they're very big, and you need to make space for them. So in this example over here, very, very condensed chromatin right here. These remodeling complexes come in, and they're represented in the bluer things right there, the ATP-dependent remodeling complexes, and it opens the DNA, and the polymerase can come right in and start transcribing the gene. In the process, though, it makes something called an mRNA sequence, which is a mirror image sequence of the DNA. This is not that. Say this is an A, that has to be a T, and it can't not be. We have the DNA in here, right? and the remodeling complex opened up a space for the gene to be copied. The gene was copied, an mRNA sequence was made all in here and then shipped out here. It ends up meeting up with a ribosome. The ribosome is going to help synthesize that mRNA sequence into a protein. And proteins are a lot of times enzymes. Actually, most enzymes are proteins. Not all proteins are enzymes though. There's a ton of proteins that aren't enzymes. They call heterochromatin the denser form of DNA, and the euchromatin is the less dense. Ready genes that are being copied all the time, genes involved with growth, root growth, photosynthesis, things that are needed all the time. They're always in the euchromatin state, so they're always being copied. Maybe a flowering plant that's not in flower, genes for flowering might be heterochromatin, they might be more condensed. That would just help make room within inside this nucleus for all these genes to fit in there. One of the mechanisms it has. From this, there's an mRNA strand made. Is a mirror image copy, like I said, and the ribosomes are going to start making your amino acids. So there's the mRNA meeting up with the ribosomes, which are green. You can see how the 3 sequences in the mRNA sequenced for 1 amino acid. So the 3 oranges, 1 amino acid, 3 yellows, 1 amino acid. And those are making polypeptide chains, which are then transported to other parts of the cell, depending on their signal sequences, which are usually right at the beginning of a polypeptide. So the first couple sequences that are involved actually signal growing polypeptide where to go. So either import into the ER, import into the mitochondria, import into the nucleus. There are these certain signals. So you can see lysine, 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 arginine, lysine. That means import into the nucleus. That protein that was coded for, that was even made in the nucleus, sent outside of the nucleus, coded for in here, sent out here. Now this ribosome's like, oh, it needs to go back into the nucleus, so it goes right back into the nucleus because of this signal sequence. Now say that there was the mitochondria, the arginine separated by a couple more amino acids, so arginine, 3 amino acids, arginine, lysine, arginine, arginine, proteins made in the cytosol. Some are imported in the nucleus, chloroplast, mitochondria, peroxisome, and then the ER, which I haven't really explained yet. The ER is a mechanism for excretion out of the cell. When proteins are made that aren't needed in the cell, they're put into the endoplasmic reticulum, which you remember me just talking about. These ribosomes actually associate with the endoplasmic reticulum, take the mRNA, and have it go through, and they're making these proteins. So here we go, here see it's all folded up inside this endoplasmic reticulum, which is right here. So these ribosomes, they looked a lot bigger in that other picture, I know, but the proteins get injected into this little white space in here inside this googly thing. Now this is where they get modified, and these proteins are going to get modified, and into this white space it gets excreted over here, and it gets modified in here. Like there's sugar groups that can be put onto it, phosphate groups, and then it gets excreted into the Golgi apparatus, and the Golgi can actually excrete outside the cell. This is the Golgi apparatus right here in red. That's a vesicle, and inside that, if proteins needed in maybe another cell. Figured that would probably be as far as I go. So we have the proteins that are now involved with moving from the nucleus outside into the cytosol, being signaled to where they need to go, and now the proteins can be functional within inside that organelle. And we'll talk about specific functions of the organelles in another video. We'll start going into the exact function of the chloroplast, how the chloroplast absorbs photons of light. And it's very, very interesting. And we can also go into how the roots absorb different particles of nutrients and stuff like that, how it breaks it down, how mycorrhizae has a symbiotic relationship with the roots. It also can have a bad relationship. If you have too much mycorrhizae, the mycorrhizae can actually eat a little bit more water than you want it to. So all stuff we'll go into, and I hope you guys enjoyed the first episode, and take it easy. Peace out.